can be serotyped by the presence of at least two structure proteins, which is a VP4. So the type or the serotype that contain or classifying VP4 known as the G types. At least there's an 8 serotypes of this pattern. And VP7, via protein 7, known as a P type, at least it can be classified to 11 serotypes. That's why the virus is quite variable, and the common strains that cause human infections, known as P8G1, P4G2, P8G3, and P8G4, try to remember these patterns because these are or can be used to design the vaccine for the rotavirus. Other family known as Adenoviridae, so genus known as adenovirus, at least there is a 50 serotypes for this genus affecting human, and serotype 40 and 41 can result in a viral gastroenteritis. So it's also non-enveloped, measuring 70 to 90 nanometer, containing a capsid, so icosahedral, encompassing double-stranded DNA, and as you can see, like a long fibers protruded from the nucleocapsid. Other group known as Astrovirus. So Astro means star. So the nucleocapsids may look like a 5-pointed pentagram. So very small, 28 nanometer, and is non-enveloped and the capsid of an icosahedral symmetry encompassing single-stranded RNA of positive sense. The last group known as the Kalisi viruses. So Kalisi means cup. So genus like norovirus and saporovirus. So small, and it looks like a cup shape, and the capsid is an icosahedral, but under the microscope it may look like the David star. Okay. Also the encompassing single-stranded RNA of positive sense. The rotavirus, which contains 7 groups, you have a group A. Is there any problem? Can you hear very well everything?
Yes.
Fine. So rotavirus, at least it has 7 groups. Have a group A, rotavirus, so resulting in infantile gastroenteritis, and the vaccines that are designed to prevent this kind of infection usually targeting this group. Group B may result in a limited outbreaks of adults. Gastroenteritis may be in India and China, and group C also minimum sporadic and outbreaks in children, and the other groups only affecting animals. But the group A, rotavirus, really result in what we call it a severe infection, especially in children less than 5 years. The pathogenesis, the virus firstly have to attach by the VP4, VP7 surface proteins. Remember these two proteins that define the virus serotypes, and they penetrate the cells of the brush border lining of the gastrointestinal system, especially the small intestine, through the receptor-mediated endocytosis, as you can see from the diagram. And the virus will lose its capsid, and the single-stranded RNA, which can act as the messenger RNA, and it will form what we call it the viroplasm. Okay, so it's like a location or a perinuclear large compartment where the viral replication and assembly will take place, known as the viroplasm. Then the virus RNA will get packaged with the double-layered capsid, and then the virus will be released from the cells, and the cell actually will get lysed, so resulting in death of the cell. In addition, the virus non-structuring protein known as NSP4 may act like an enterotoxin. That's to say, it may result in what we call it, or prevention of a reabsorption, by inducing calcium ion dependent chloride secretion, and in addition to the blockade of the transporter mediated reabsorption of water. And this is result in a massive destruction of the cross-border cells and malabsorption phenomena. So the reabsorption will get interrupted, in addition to the secretion of the water from the inside to the outside. So the transmission is mainly fecal-oral transmission. That's why it's very common in areas with a poor sanitation and lack of clean water supply. That is why it's quite common in Africa and South Asia, notably India, and also Eastern Africa, like in Sudan. Incubation period is about 3 days, and that's why it's quite the incidence is quite common in children, and may result in one of the serious cause of dehydration among them. The deaths related to the rotavirus gastroenteritis among infants may reach up to 1 million per year. To diagnose the rotavirus in the lab, so we can do antigen detection by ELISA, or we can use what we call immune electron microscope. And as you can see, so virus may look like a wheel under the microscope, especially if you enhance through an immune attachment maneuver. And also we can use PCR, in this case RT-PCR, for the viral nucleic acid. The treatment, so the only way is to make the child hydrated as much as you can. So if in case of mild rehydration, you may just use oral rehydration solutions, or in severe rehydration, the child may need admission and a massive IV fluid replacement. There is no antiviral treatment for this infection. However, this infection can be prevented through vaccination. One of the old vaccines, known as the RotaShield vaccine, which is a tetravalent live attenuated vaccine. However, this vaccine right now does not exist because it was withdrawn due to the association with intussusception among children. The newer vaccines, so they have a monovalent vaccine, like Rotarix vaccine. It's a live attenuated vaccine. However, it's only monovalent because it prevents from PAG1 subtype. It has given 2 doses, but the main problem is not effective against the other serotypes. The best one these days is a pentavalent vaccine, like a Rotatec vaccine, and it contains the 5 strains. And this is produced by the genetic reassortment and genetic manipulation, and contain the most famous or the most common subtypes like G1, G2, G3, G4, and G6, and is given in 3 doses. Few words with regard to the Norwalk virus, or the norovirus, which is a Kalisi virus, which is discovered by accident in 1972 during the outbreak in a city in the United States known as Norwalk. It affects all age group, and it is associated with epidemic form or the outbreak, especially in schools and hospitals. The incubation period is also short, like 72 hours, and usually the patients will develop explosive vomiting, may be accompanied with diarrhea and abdominal pain. The transmission can be oral fecal route through contaminated food and water, and it can be through person-to-person transmission by aerosolized projectile vomit. Finally, there's other causes of gastroenteritis caused by adults, like by viruses, like astrovirus, can result in outbreak of gastroenteritis in school and children, and adenoviruses result in like less than 6% of infantile gastroenteritis. However, the severe cases can occur in immunocompromised, especially HIV and post-transplant individuals. The main preventive tools is to improve hygiene through proper hand washing and sanitation and provision of a clean environment and water supply and safe drinking water and food. So just remember the term for before you drink or eat. You need to boil it or cook it or peel it, or if you can't do that and are not sure that this is a clean food and water, just forget it. Some textbooks for further details, and thank you very much. Is there any question? Is there any question? Yes, brush border cells. Okay, these are the cells within the, especially the GI, small intestine mainly, because the main role of the small intestine is absorption of fluids and water, because it contains something we call it microvilli. I think people in histology and anatomy, they talk about that in much more details, because it will increase the surface area for effective absorption of fluids. If these cells are dead, the brush border cells in the GIT, especially in the small intestine, are dead, the water and fluids cannot be reabsorbed. That's why the patient will develop what we call malabsorption and will develop massive diarrhea. Is there any question? Any other question?